Now let's look at the function of each part from the mouth right to the anus. Okay, one by one we'll look at the function of each component. Now the first one, let's look at digestion first. How does digestion occur in each part of the alimentary canal? First of all, let's look at digestion in the mouth. Now in the mouth, starch will be acted upon by salivary amylase, also called as ptyalin. P is silent, clear? It's also called as ptyalin at a pH of about 7.5, and it will change into maltose, which is why if you chew some cooked rice for a long period of time, the rice starts to become sweet. Initially it will have no taste because starch is insoluble and it's tasteless. But once it mixes with the saliva, the amylase will convert the starch into maltose, and maltose is slightly sweet. So the rice will start to taste sweetish. Is it clear? Now no further digestion takes place, but the teeth will break the food into smaller pieces, and what does that do? And that increases the surface area for enzyme action. So enzyme action will increase when the surface area is more. Is this clear? So the food is then made into a bolus and swallowed. Now in the esophagus, the same thing will happen. There will be the continuation of digestion which begins in the mouth till the food reaches the stomach. So here also starch changes into maltose by salivary amylase. No other change. Is it clear? Then the food then goes into the stomach. Now as soon as the food goes into the stomach, the salivary amylase is deactivated. So in the stomach, first of all, the low pH due to hydrochloric acid, pH of about 2 to 3, what does it do? It deactivates salivary amylase. And now digestion of starch will stop. The proteins in the food will be broken down into polypeptides by an enzyme called pepsin, which is a protease enzyme, and it works best at a pH of about 2 to 3 because of HCl in the stomach, hydrochloric acid. No further digestion will take place. The food will be churned and then slowly let into the duodenum. Now, just for your information, just to make it a little easier, see, these are what proteins are. Proteins are made up of long chains of amino acids. And when we talk of the proteins being broken down into polypeptides, what we mean is that this long chain of proteins, by the breaking of some bonds, they'll be broken into smaller chains. That's why we say the protein is changed into polypeptides. Is this clear? So this is what happens. But it's still made up of many amino acids. That's why they are still called as polypeptides. Next, the food now goes into the duodenum. And into the duodenum, two very important structures will pour their secretions. One is bile from the liver, and the other is the pancreatic juice from the pancreas. Now the bile has no enzymes, but the bile has sodium hydrogen carbonate, which will neutralize the HCl. So it neutralizes hydrochloric acid from the stomach, and it now makes the pH alkaline, somewhere about 8 to 8.5. Clear? And the pancreatic juice has three enzymes. It has an amylase, pancreatic amylase, it has a lipase, and it has another protease called trypsin, which is another protease. A protein digesting enzyme is called as a protease. Is it clear? Now, what does the bile do? The bile emulsifies the fats. We'll look at that later. Now, the same enzyme will now act upon starch. See, starch, which was not completely digested in the mouth and the esophagus, will now continue its digestion with the help of the enzyme pancreatic amylase at a pH of about 8 to 8.5, and it will change into maltose. Now, just for your information, see, starch is a chain of glucose. Now, glucose has this hexagonal shape, and many glucose molecules are joined together to form starch. Is it clear? Now, when the starch is broken down into maltose, maltose is a pair of glucose molecules. So now the glucose will be broken down into pairs, or the starch will be broken down into pairs of glucose molecules, which we refer to as maltose. Is it clear? Then lipase. It will convert lipids. Now remember, a lipid is made up of one glycerol joined to three fatty acid tails. The curly ones are the fatty acid tails. and the lipids will be acted upon by pancreatic lipase at the same pH to form fatty acids, which are those curly ones, and glycerol. Is that clear? To which the 3 fatty acids were attached. And finally, you have polypeptides, and remember the polypeptide was a long chain of amino acids. You remember? It got a little shorter in the stomach, but not completely broken down. Now these polypeptides, with the help of trypsin with a pH of 8, will be broken down into dipeptides. Now dipeptides are pairs of amino acids. So you see, it's all broken down in stages. Is that clear? And what does bile do? See, bile will emulsify the fats. In other words, if this is a big fat globule, when you add bile to it, it still remains as fat. Chemically, it doesn't change, but it's broken down into many small droplets of fat. Is that clear? So this is emulsified fats, which means that they have a larger surface area for the enzyme lipase to act upon. So any doubts on what happens in the duodenum? Then the food next goes to the ileum. Now in the ileum wall will secrete its own enzymes called the intestinal juice, which is secreted by the walls of the intestine itself. And this again has 3 enzymes. One is maltase, M-A-L-T-A-S-E. The other is erepsin, which is also a protease, and the last one is again lipase. Clear? Now here the process of digestion will be complete. Now you remember the maltose which formed in the duodenum. The maltose will be acted upon by the enzyme maltase, and it will be changed into glucose. In other words, the pairs of glucose molecules which were paired up will now be separated, and now you'll have individual glucose molecules. So digestion is complete. The long chain of starch which had many glucose molecules joined up, now it's completely broken down into single units of glucose so that the glucose can be absorbed into the blood. Clear? Secondly, the dipeptides will now finally, this pH 8, with the help of erepsin, which is also a protease, at the pH 8 will be changed into amino acids. Now remember, dipeptides had this structure. Now they'll be broken down into individual amino acids which have no link with each other. And these can then be absorbed into the blood and then taken to the cells and used to make our own proteins. Clear? And then finally you have the lipase, which will break down the remaining lipids which couldn't be digested in the duodenum with the help of the enzyme lipase, same pH of 8, and it will change into fatty acids plus glycerol, which can then be absorbed into the lymph vessels. They don't get absorbed into the blood. They'll be absorbed by the lymph, taken into the blood later on. Okay, so this is the lipids which have 1 glycerol and 3 fatty acids. Now the fatty acids and the glycerol will be separated from each other. Is this clear? So this is the complete process of digestion which occurs.